Good evening, class. Let's now see how we can effectively measure plasma or blood glucose level. Glucose, as earlier on discussed, is a carbohydrate whose most important function is to act as a source of energy for the human body by being the essential precursor in the synthesis of ATP, also called adenosine triphosphate. The energy stored in ATP can then be used to drive processes requiring energy, including biosynthesis, locomotion, or transportation of molecules across cell membranes. This is our glucose. This is a glucose unit. So we represent this as a glucose. Many of the foods we eat are broken down during digestion to this simple sugar, glucose. Glucose, therefore, is carried to every cell in our body by the bloodstream, where it is used as a source of energy. Glycogen is a stored form of glucose. Glycogen is made up of many connected units of glucose. So if we have one glucose here, glycogen will be connected. Many glycogen comprises of what? Many connected units of glucose. What is insulin? Insulin is a hormone released into the blood when blood glucose levels are low. It enables glucose to be released from some tissues back into the bloodstream. Insulin is a hormone released into the blood when blood glucose levels are high. It enables glucose to be transported into the cell in some tissues. So whenever blood glucose is high, the beta cells of the islets of Langerhans, beta cells of islets, ISLGT, of Langerhans produce insulin. Whereas the alpha cells of islets of Langerhans produce glucagon. So note the differences. The beta cells of the islets of Langerhans of the pancreas produce what? Insulin. Whereas the alpha cells of the islets of Langerhans of the pancreas produce glucagon. And insulin is what mobilizes or sends plasma or blood glucose, if it is in excess, goes to store them, causes the storage of plasma glucose or blood glucose in the liver, in the muscle cells, and the fatty adipose tissues. Whereas when we are fasting, or when immediately we need an energy, with immediate effect we need an energy during when we have been attacked by armed robbers, rapists, and whatever, we need to mobilize a quick source of energy. So glucagon is a hormone that is produced by the alpha cells of the islet of Langerhans, of the pancreas, and the glucagon will now break glycogen down into glucose unit, which will now be broken down through glycolysis to release pyruvate. Pyruvate will also release, glycolysis will release 2 ATPs. It will go through Krebs cycle to release some energy. It will also go through the oxidative phosphorylation to release about 34 units of ATPs. So glucagon, this is a hormone released into the blood when blood glucose levels are low. It enables glucose to be released from some tissues back into the blood. The body organs. Pancreas, one of the major players in glucose homeostasis or glucose regulation. The pancreas releases the hormones insulin and glucagon that control blood glucose. Insulin is released from what? Beta cells of the islet of Langerhans of the pancreas, whereas glucagon is released from the alpha cells of the islet of Langerhans of the pancreas. The liver is also an organ that takes up glucose when levels are high and releases glucose when levels are low. So it's a storage site. It stores glucose in chains as glycogen. It is key for glucose regulation. Our muscles are able to take up and store lots of glucose when insulin is present. More muscle mass means more of a reservoir for glucose. So if you are fat, it means you have a lot of glucose reservoir. If you are lean, then trouble for you. Fat cells. Fat cells take up glucose when insulin is present. Fat cells use glucose to make more fat. The brain takes up glucose whenever it needs energy and doesn't require insulin. So the brain does not require insulin to take up glucose. Glucose is the fuel the brain normally uses. So let's look at this diagram. All of these systems work together to keep our blood glucose level balanced. So this is a balanced glucose level. You can see a lot of glucose here and balanced blood glucose. The glucose level of the blood is balanced. You can see the seashore. Okay. High blood glucose triggers the pancreas to release insulin. So you can see the glucose is so much, very, very heavy in the blood that the seashore has gone down and the high blood glucose is up. So the pancreas now releases insulin. Insulin goes into the blood, binds to glucose, and takes them for storage in the liver, muscle cells, and the adipose tissues. Again, when glucose level is low in the blood, we call it hypoglycemia. Hypoglycemia. Low glucose concentration in the blood. Hypoglycemia. Low glucose concentration in the blood. The opposite is hyperglycemia. That's high glucose concentration in the blood, hyperglycemia. So, when there is hypoglycemia, blood vessels carry glucagon to the body to trigger the release of stored glucose back into the blood. So you can see blood vessels, glucose is in blood vessels. So the pancreas, the alpha cells of the islet of Langerhans of the pancreas will release what? Glucagon. And the glucagon will what? Go to the blood vessels. Then it will trigger the release of stored glucose back into the blood, so that the body can now use it for its normal metabolic activities. Okay, so glucose is in a balance. Glucose is now balanced in the blood. This balancing act happens many times a day. Every time you have a meal or consume a drink with sugar, the ability of the body to maintain balance and regulate internal condition is called homeostasis. Diabetes mellitus. Diabetes mellitus is a chronic disease caused by inherited or acquired deficiency in production of insulin by the pancreas or by the ineffectiveness of the insulin produced. Such a deficiency results in increased concentrations of glucose in the blood, which in turn damage many of the body's systems, in particular the blood vessels and nerves. Diabetes can be classified into the following categories. Type 1 diabetes. This is also called autoimmune beta cell destruction. That's the islet of Langerhans, the beta cells of islet of Langerhans are being destroyed by the body. So for instance, if you have the pancreas, so this is the pancreas divided into two. We have the alpha cells, we have the beta cells. And this whole structure is the what? Pancreas. So the pancreas, we have the alpha cells and the beta cells. So if the beta cells are being destroyed by chemicals released by the body, example cytokines, interleukins, if the body releases them to destroy the beta cells, then we have type 1 diabetes. And this is what happens in children, people of a younger age, and they are having diabetes. Young children, they are having diabetes. It means that their own body components, their innate immune system is fighting against their beta cells of their own pancreas, giving them diabetes mellitus type 1. We have type 2 diabetes. This is an adult onset diabetes. It's for those who are older, the aged, like you are from 40 years going, 35 years going, due to a progressive loss of beta cell insulin secretion, frequently on the background of insulin resistance. So when there is resistance to insulin, the person is producing insulin alright, but the insulin is not able to mobilize glucose into the liver and the body cells, adipose tissues, the muscles. We don't know what is happening. There is what? Insulin is being produced in normal quantity, but it's being resisted. It's not able to do its work. Gestational diabetes mellitus. Some pregnant women when they are pregnant, when they are not pregnant, they don't have diabetes. Their sugar level is normal. But when they are pregnant, their sugar level goes up. Why? And it happens that when they finish giving birth, their sugar level becomes normal. This condition is called gestational diabetes mellitus. Gestational means what? Anytime you hear gestation, is what? Pregnancy. So pregnancy associated diabetes mellitus. We have specific types of diabetes due to other causes. Disease of exocrine pancreas such as cystic fibrosis and pancreatitis, that's infection or inflammation of the pancreas. Drug or chemical induced diabetes such as glucocorticoid use in the treatment of HIV or AIDS or after organ transplantation. These are the symptoms of diabetes. The person feels tired, that is fatigue. The person urinates a lot, especially in the night. You can wake up about 5 times or 10 times to urinate once he didn't take in much water. The person has a lot of infection because if you have sugar in your blood, it will invite bacteria and other parasites. Small time they are hungry, they can eat right now within 30 minutes or 10 minutes and they are hungry because the sugar is not going anywhere. It will just come into their urine. They will just urinate all the sugar. They have what? Neuropathy or diseases associated with the nerves. They have nephropathy, that diseases associated with the nephrons or the kidneys. And the nephron is the functional unit of the kidney. They have heart problems, coronary heart problems. They have stroke, retinopathy, peripheral vascular disease. They easily get thirsty. They have vision problem. They are not able to see well. So these are symptoms of diabetes. That is, if you are diabetic and your sugar level is not controlled, this is what happens to you. But if your sugar level is controlled, you don't have any problem. You will not encounter any of these symptoms. By measuring blood glucose level, how do we measure blood glucose level? Fasting glucose test. After fasting for at least 12 hours, a person's blood is drawn and tested for glucose. A healthy person would have a fasting blood glucose level of about 70 to 100 milligrams per deciliter. So fasting, they will say don't eat in the evening around 6 or 7 after you have eaten. Don't eat again, but you can take in your drugs. The next day, you do the test. Exactly 12 hours. If your last meal was 6 o'clock in the evening, then in the morning by 6 o'clock you should take the test because 12 hours. Oral glucose tolerance test. After measuring fasting glucose, a person is given a glucose-rich drink, and this is normally for the pregnant women who have gestational diabetes. Blood is then drawn at time intervals to see how that person's body is processing the glucose. Fasting 60 to 100 milligram per deciliter. So what happens is that the person would go through this process, the fasting blood sugar test. Then you give the person about 750 milligrams of what? 750 grams of glucose that has been diluted in this in mineral water. Then you mix it and give him her to drink. After drinking, 30 minutes time you do the test. 1 hour time you do another test. 1.5 hours you do another test. 2 hours you do another test. And these are the reference ranges that you expect. After 1 hour you should get less than 11.1. After 2 hours you should get less than 7.8. For diabetes testing, hemoglobin A1C test or glycated hemoglobin is the best. Is the gold standard. Hemoglobin A1C is the gold standard for diabetes testing. The most important test. the gold standard. Here we look at hemoglobin of the blood that has been married or conjugated with what? Married or bounded or conjugated with what? Glucose. So that's the best test, and the reference rate is between 4 to 6.0 or so. The reference rate is between 4 to what? 4 to 5.6. Okay, 4 to 5.6. 4 to 5.6. 4 to 5.6. That's the reference range for glycated hemoglobin testing. Why do we monitor blood glucose? It reduces the risk of developing complications with diabetes, blindness, such as, so if you monitor your blood glucose and it's always normal, you will not get blindness, kidney failure, coronary thrombosis, that's thrombus or a plaque being found in the coronary veins of the heart. We have the stroke. We have also bacterial infections and fungal infections. When your glucose level of your blood is controlled, it allows to see if the insulin and other medications that you are taking are working. So if you monitor blood glucose level by testing, it allows you to see if the insulin and other medications that you are taking are working. Because those with the type 1 diabetes, most of the time they inject their body with insulin. Frequent testing gives diabetics an idea as to how exercise and food affect their blood sugar. May prevent hypoglycemia. If you don't test, you may not know that the drug that you are taking has totally reduced your blood glucose level, which is also a danger. Or your blood glucose level has shot up, which is also dangerous. If you are hypoglycemic, it means that you are very hungry, you don't have any glucose in your blood, so you can end up fainting or collapsing anytime. Hyperglycemia too is accompanied with this complication: blindness, kidney failure, coronary thrombosis, stroke, bacterial infection, and a whole lot. So in a nutshell, the sugar level should be controlled in a diabetic. So you can see fasting blood sugar. A person without diabetes, you are supposed to get 3.9 to 5.5 millimole per liter. Please, the unit millimole per liter is what is common in our setting. So most of the time, most of our machines have millimole per liter as their unit. So 3.9 to 5.5. Then for somebody with diabetes, the fasting blood sugar test is 7.8. Then after 2 hours postprandial, 2 hours postprandial or glucose loading, that's postprandial. That's when you finish eating and you test your blood sugar level after 2 hours. We call it 2-hour postprandial test. 2-hour postprandial. 2-hour postprandial glucose test. So after 2 hours, this is what we should see. We should see that for somebody who is normal, somebody who is without diabetes or normal, it should be 7.8. So we say it's like a random blood sugar test. Random blood sugar test is when you are finished eating and you test. It's not supposed to be more than 11. So here 7.8. But somebody with diabetes, after eating 2 hours time when you test, it's supposed to be more than 11. millimole per liter. How do we test? We have methods. We have the oxidation reduction methods. Example, Falling-Wu, Benedict's method, Somogy-Nelson. Condensation methods, that's the Otto Tolodin. We also have glucose oxidase. The glucose oxidase test is what I did when I was doing my undergraduate project in the year 2006. Hexokinase, glucose dehydrogenase. So these enzymatic methods, we use ELISA technique. ELISA means what? Sorry, ELISA, ELISA. It means enzyme linked immunosorbent assay. This is what we use to test for the concentration or measure the concentration of the various enzymes over here. So we have ELISA test. Let me clean this E and write it properly. ELISA test. It's also called enzyme link immunosorbent technique. Enzyme link immunosorbent technique. So ELISA. So we use ELISA test to measure this concentration of these enzymes. This is the glucometer. I guess you all know how to use the glucometer. We'll go, and when you go to the hospital, you'll learn it. So glucometer is an electronic device used to test the amount of glucose in the blood. New models are able to read and even calculate the blood sugar within seconds. Some models not only display the glucose reading but also mention it, or they say it. So this is how it is used. Step 1, put the strip inside the glucometer. It's like the GCSPD biosensor, the same principle. Prick side of clean finger tip. Press finger to get more blood. Touch drop to test strip in meter. Read blood sugar results in meter. Thank you very much. Now we will be going for our last lecture, which is on reducing sugars.